Hello, everyone. My name is Veronica Martinez, and today we're going to be discussing bacterial resistance against beta-lactam antibiotics mediated by Class A beta-lactamase. All right, so how prevalent is antibiotic resistance? Antibiotic resistance kills about 1.27 million people worldwide. Antimicrobial resistance occurs when bacteria develop the ability to defeat drugs that are meant to kill them. Resistance may occur naturally, so it makes it even harder to completely stop or minimize their effect. Being able to find a way to minimize antibiotic resistance is needed because antibiotics have saved many patients' lives or just about anyone. And they play a pivotal role in changing advances in medicine. All right, so there's three main mechanisms of resistance to beta-lactam antibiotics. These three main mechanisms are: one, the production of beta-lactam modifying or degradative enzymes; two, target modification; and three, the regulation of beta-lactam entry and efflux. For this presentation, we will be focusing on mechanism number one, which is the production of beta-lactam modifying or degradative enzymes. Right, so in the last slide, I mentioned beta-lactam antibiotics. So what exactly are beta-lactam antibiotics? The first beta-lactam antibiotic discovered was in 1928 by Alexander Fleming, and is what we know as penicillin. Since the discovery of penicillin, it has been widely used and is very effective at treating infectious diseases. It is a common antibiotic that is constantly prescribed to you when you go to the doctor with an infection. They actually obtain their name due to their structure, which is a four-member lactam ring. As you can see over here, we have a little arrow here that says that is a four-member lactam ring. They treat bacteria and infections by inhibiting peptidoglycan biosynthesis, or just the SOA synthesis. Specifically, they target the nucleophilic serine of PBPs, or penicillin binding proteins, which leads to the inactivation of which leads to the opening of the ring of that beta-lactam ring, and it prevents the formation of peptidoglycan transcripts. All right. So now, I previously discussed a little bit how they do this. They work by inhibiting SOA synthesis. Now we're going to go over a little bit, just briefly overview of SOA synthesis of bacteria. So we know that the main function of a SOA is to maintain shape and withstand intracellular pressure. We have also learned that there's two kinds of bacteria, SOA, gram-positive and gram-negative. The gram-positive contains that thick layer of peptidoglycan, while the gram-negative contains a thin layer of peptidoglycan. As you can tell in these two pictures over here, we can see the difference. And the gram-negative has that extra outer membrane layer. So what exactly is peptidoglycan? Peptidoglycan is a combination of glycan plus peptide crossings. Now, glycan are sugars known as NAG, or N-acetylglucosamine, and NAM, N-acetylmuric acid. NAG and NAM are essential building blocks of the cell wall. So now we're going to talk about the three main stages of the cell wall synthesis. So in the first stage is where NAG and NAM, the sugars that make up glycan, are synthesized. And then we have two, the membrane stage. This is the stage in which the building blocks are put together. And then lastly, we have extracellular stage. And this is is the cross-linking of linear chains of peptidoglycan by membrane-bound transpeptides. Transpeptides, or PPPs, is an enzyme that creates crosslinks between amino acids. The extracellular stage is a stage in which antibiotics attack. This is the stage where they belong. This is where they do their damage in order to be able to stop the bacteria or infectious disease. As I mentioned before, the mechanism of resistance of beta-lactam antibiotics is the production of beta-lactam modifying or degradative enzymes. These enzymes are known as beta-lactamines. They have existed prior to the discovery of nature, but were found in a lab in the 1940s. Many scientists actually believe that they evolved from PPPs or penicillin-binding proteins. Not only that, but they can be grouped into 4 kinds of beta-lactamines: class A, B, C, and D. Classes A, C, and D are of active site serine enzymes, while class B are heterogeneous groups of zinc metalloenzymes. So that just means that their active site is a metal zinc. Class A enzymes work by inactivate antibiotics by using a serine as a reaction nucleophilic and then hydrolyzing the beta-lactamide via covalent acioenzyme intermediate. And for this PowerPoint, we're going to be looking more into depth on class A beta-lactamines. So class A beta-lactamines, they exist in both gram-positive and gram-negative bacteria. They are known to use a mechanism which involves a nucleophilic attack by SIR-17 on the carbonic carbon of the antibiotic. After this, an acioenzyme intermediate is formed. They're widely distributed and extensively studied beta-lactamines. They're probably one of the most studied beta-lactamines out there. And some of the most known beta-lactamines are PC1, TEM, SHV, CTXM, and KPC. TEM1 similar is a common A beta-lactamines, and it can hydrolyze penicillins in early generations of cephalosporins. So despite everything that is known about class A beta-lactamines, there's still debate on how exactly the acylation mechanism occurs. So beta-lactamines work in an acylation-deacylation mechanism. There's 2 big steps: the acylation and deacylation. The deacylation portion of this mechanism is known, but the acylation portion of it is not known. So there's 2 proposed general bases: glutamic 166 and lysine 73. What they're trying to find out is which one acts as a general base that activates SIR-70 during acylation. It has been in debate for a while now, and they have used many structural and X-ray crystallography studies to confirm which one it is. They have found that when lysine is pronated, the substrate binding recognizes pronation in the active site, and in turn, lysine 73 activates SIR-70. But when glutamic 166 is depronated, the serine 70 is activated by a nucleophilic attack upon the beta-lactam carbonyl, and by this in turn, glutamic 166 via water molecule activates the SIR-70. All right, so now we're going to talk about the actual acylation and deacylation mechanism. So acylation occurs in 2 steps: the formation of the tetrahedral intermediate, and 2, the formation of the acioenzyme. Deacylation is when the conserved water molecule is activated by glutamic 166 to hydrolyze the acioenzyme intermediate. So in order to understand this better, best if we look at pictures of them. So in A, we have structure number 1. And structure number 1, first let's start by in structure 1, 2, and 3, this is all alkylation half of the reaction. 4 and 5 are the dealkylation. So in 1, it shows the ground state Michaelis complex. In 2, is how the Michaelis complex then proceeds through a high energy alkylation transition state. So this is a transition state. And then we have 3. From the transition state, it then goes to an active enzyme intermediate. So this is where the intermediate is formed. And as you can tell from the pictures, we start off with the beta-lactam ring being closed off. And then it proceeds, the SIR-70 then attacks it, and then the beta-lactam ring ends up being opened. And then in the bottom, we have, let me finish. Okay, so structure number 4 is a dealkylation process through a high energy dealkylation transition. And then lastly, we have the post-covalent product complex that then ends up leaving. In the bottom at B and C, we have the 2 mechanisms that I talked about before, the 2 proposed mechanisms, glutamic 166 and lysine 73. And it's just showing a possible mechanism of how they attack the beta-lactam antibiotics. So we're not going to go over the nucleophilic attack and formation of tetrahedral intermediate when glutamic 166 is the general base. So in the figure to the right, there's 3 steps, X, Y, and Z, and they're with the arrows. So in event Y, a proton is abstracted from SIR-70, and via bridging water, a second proton is transferred to the general base glutamic 166. The deprotonated SIR-70 attacks the carbonyl group of the electron ring, which then causes it to open. So this is just how the nucleophilic attack happens, which then in turn forms the formation of the tetrahedral intermediate. Glutamic via a bridging water, and then that water with proton, and then it attacks the carbonyl group of the electron ring. And how it produces an electron density map. A crystal is used to build a 3-dimensional molecular structure or protein of a protein or biological macromolecule. An electron density map is a plot that shows where in space electrons are concentrated within the repeating unit of the crystal. Now, this process is started with the purified sample that is crystallized and then exposed to an X-ray beam. After it has been exposed, it will produce diffraction patterns. These diffraction patterns tell us about crystal packaging symmetry and the size of the repeating unit that forms the crystal. The diffraction spots have intensities that can be used to determine the structure factors, which in turn creates the electron density map. This is how the electron density map is created, by these diffraction patterns and spots that are intensified. When these X-rays are done in a high resolution, it can determine the pronation states of the 2 proposed bases. All right, so to the picture on the right is showing in an electron density map. And we can see here the hydrogen bonding in streptomyces strain R61. This is sort of what they look like. Most of them look like. Some of them can go further into each atom that can be seen. All right, so for my hypothesis is that during acylation of a class A beta-lactamase, glutamic 166 acts as the general base that activates SIR-70. Okay. So in this first figure, we have oxygen. We have an electron density map that was calculated, and glutamic 166 is pronated. We know that it is pronated due to the red peaks in E166 and W1004. So let me just explain what we're seeing here. So in the blue the highlighted blue are selected residues of the active site. The red, there's little red peaks or intensities. Those indicate that carboxylic group is clearly pronated. And then here we have, if we go deep, deep into the structure, we can see like that red right there. We have carbon in yellow. We have blue as nitrogen. We have red as oxygen. We have green as sulfur, and hydrogen atoms are green. All right, so now, now that I've discussed what everything means in the electron density map, there is red spots surrounding the carboxylic group. So at the ends of the E166 right here, there is red peaks. And these red peaks, there's two ends to the E166. There is two red ends and then a yellow, which I put a figure above so you can remember what a carboxylic group looks like. It has the two oxygens. Remember, oxygen is red. So the two red ends, which are oxygen, and then an R, which the R stands for the rest, the rest of the molecule, the rest of the structure. So this confirms that the red peaks are the carboxylic group. This is actually the carboxylic group of E166. So the W1004 or the water molecule also indicates by its position that it is ready to accept a proton from glutamic 166 and donate its own to SIR 70, in turn activating it. So we can see here the E166, the water, and it shows the two red peaks, and the SIR 70 here. Remember that glutamic 166 is pronated, and via a bridge in water, a second proton occurs. The serine 70 is activated, and it attacks a carbonated group of beta-lactam ring. Overall, these will then lead to a proton shuffle, which we're right. So now we're going to be discussing potential energy differences during pronation of glutamic 166. In this slide, we're going to talk a little bit about potential energy differences and the methods used to create this figure. So quantum mechanics and molecular mechanics methods were used to calculate the potential energy differences. Additionally, they also used the events I described earlier in the presentation as X, Y, and Z to do the calculations. This is why we have axes that have Y, Z, and X. The R, like if I say R, Y, Z, or RX, the R just stands for reaction. So in this figure, the reaction coordinate Rx or event X, which is the second proton transferred to a general base glutamic 166, showed a change in energy when glutamic 166 was pronated. So here we can just see that when glutamic 166 was pronated, the potential energy difference were changed, meaning or confirming that glutamic 166 was pronated. And then in A, this is a method called AM1CHRMM22. And then in B, there was a method used as B3LYP6-31G plus D divided by AM1 CHRMM22 surfaces. These are just two different methods that are used, which help calculate and make a more concentrated and specific structure. All right, so moving on. Now we have another potential energy surface, but this time it's the step of the acylation, and with the addition of the reaction energy profile. All right, so the many studies have shown, or in this case, this in this figure, we're going to see that the acylation step follows a concerted reaction mechanism, where activation of SIR-70 and nucleophile attack happen at the same time or simultaneously. In one here, we can see the Michaelis-Menten complex. Two, we have the hydronium ion structure. This structure was specifically used because it resembles the geometry of glutamic 166. And third, we have the tetrahedral intermediate after glutamic 166 is pronated. So in square number 3, it was concluded to be more stable than the Michaelis-Menten complex in square number 1. Sorry about that. So the Michaelis-Menten complex is way more. No, the tetrahedral intermediate is more stable than the Menten complex. All right, so in the reaction energy table, this graph up here, sorry, it also agrees with the theory that that events X, Y, and Z happen united. This is known due to the structure number 2, so the hydronium structure. It shows here. It shows a very small barrier of less than 1 kcal/mole. So all this means is that sorry, that events X, Y, and Z all happen at the same time when glutamic 166 acts as a general base. Sorry. All right, lastly, we're going to be talking about QM/MM energy profiles for acylation mechanism during tetrahedral intermediate formation that support the glutamic 166 as a general base. So in this slide, we have in the bottom of the graph, we have energies. These energies come from the MC, TS1, TI, TS2, TI2, TS3, and AE. So TS1 stands for transition state of formation of the tetrahedral intermediate. Not only that, but all the solid lines show the AM1 CHRMM22, and the dashed lines show B3 LYP, and these are just different methods used. So the activation energy for cleavage experimentally has been found to be between 12.7 and 13.7 kcal/moles. In the table, we're going to observe TS1, which is the activation energy for cleavage. So now that we know that the experimentally found activation energy is 12.7 and 13.7 kcal, we're going to focus on TS1 here. One of the methods, the AM1 method, showed a 19.6 kcal/mole, and the B3 LYP, the dash one, shows an 8.7 barrier. The B3 LYP method is preferred because the other one is usually overestimates and the B3 LYP doesn't. So for this, we're going to go with the 8.7 kcal energies. This simply supports that the mechanism in which glutamic 166 acts as the base, as a general base in acylation. This is all this is showing. It's showing the energies and how 8.7 is closer to the 12.7 or 13.7 kcal experimentally found, which is yes, since it has an 8.7, it supports that glutamic 166 acts as a general base during acylation. Okay, all right. My conclusion. So beta-lactams inactivate beta-lactam antibiotics in a two-step mechanism, acylation and deacylation. The deacylation portion of the mechanism is known and agreed upon, but not the acylation. Glutamic 166 was identified as the general base in acylation based on electron density maps in quantum mechanics and molecular mechanics calculation of potential energy surfaces. Antibiotics such as penicillin and cephalosporins are created to help kill or inhibit microbes, but they struggle to do the job when they develop resistance. Antibiotic resistance, as I said before, is a prevalent problem worldwide, and there has been ways, or scientists have created ways to help reverse this, or not reverse, but inhibit antibiotic resistance by creating inhibitors. Specifically, they've created beta-lactamase inhibitors, which is a step further into being able to solve this prevalent problem worldwide. All right, guys, thank you for watching my presentation. I hope I did a well enough job explaining what everything was. Here's my reference slide, and I hope y'all have a great day.